Presentation? Okay.
Yeah.
All right. So thank you for that introduction. And I would like to thank the organizers for the invitation to participate in this event. I'm always happy to do whatever I can to help out with promoting evolutionary biology, especially human evolution, anywhere around the world. So I'm going to start with some history, which may very well be an indication of how old I'm getting, but I do happen to think that history is important. That if we want to know where we're going, it helps to know where we've been. And so I'm going to start with this question that kept a lot of us busy for a good many years, and that is the question of how did our own species, anatomically modern humans, originate? And I want to point out that it's a bit deceptive because it makes it sound like we're interested in a single entity, a single thing, anatomically modern humans. But actually what we want to know is when we look around the world, we see what looks to us like an incredible amount of diversity in people's appearances and their skin color, hair morphology, height, weight, as well as their culture, right? Languages and other cultural traits. So we'd like to know, how did all of this diversity originate? Is it recent? Is it ancient? Is it single origin? Or is it multiple origin? Back when I was a graduate student in the 1980s, there were basically three different models. I mean, there have always been lots of different models proposed for human origins, but there are mainly three that most people were discussing and debating. And so one of these shown here is multi-regional evolution, proposed originally based on the fossil record. And according to the fossil record, everything that happened in our evolutionary past was in Africa up until about 2 million years ago. And then beginning around 2 million years ago or so, the first hominins began spreading out of Africa. And they spread across the entire old world, and they're quite successful. But what spread out at 2 million years ago wasn't anatomically modern humans, it was something else. Here it's called Homo erectus. Different anthropologists would give different names. The names don't really matter. What's important, though, is that then it's only around 350,000 years ago that anatomically modern humans appear on the scene. And so while according to multi-regional evolution, the main lines of evolution are descent within each of the major regions of the old world. So modern Africans are mostly descended from ancient Africans, modern Europeans mostly from ancient Europeans and so forth. You also have these, so that's indicated by the vertical arrows, but you also have these horizontal arrows, which indicate gene flow, which was very important then for keeping the entire old world population of hominins spread from sub-Saharan Africa to Western Europe to Eastern Asia, even out into Oceania, evolving in concert to become anatomically modern humans. Now, as opposed to multi-regional evolution, there were a variety of theories that proposed a single origin, a single place, a single time for modern humans. The most prominent of these was Africa. So again, according to the fossil evidence, you have the first hominins spreading out of Africa 2 million years ago. They're successful for hundreds of thousands of years. But then somewhere around 300 to 350,000 years ago, something special happens in Africa, and that something special is us. And then beginning somewhere between 50 and 80,000 years ago or so, our ancestors spread across Africa and they began spreading out of Africa. And in the most extreme version of the out of Africa hypothesis, which is out of Africa with complete replacement, these archaic non-African hominins are replaced completely. They don't contribute any ancestry at all to modern humans. So modern humans today are basically all African refugees, if you will. And then the third hypothesis is out of Africa, but allowing for some contribution of ancestry to modern humans from archaic humans outside of Africa. And there was debate about where this might have occurred. Was it in Europe with Neanderthals, or was it elsewhere with other archaic hominins? But the crucial difference is that it was not a complete replacement, but rather that there was some ancestry contributed by the non-African hominins. Now, these were all proposed originally based on fossil evidence, but they proved incredibly difficult to differentiate between these different hypotheses based solely on the fossil evidence. What it took was genetic evidence, because if you think about it, what these different hypotheses do is they make different predictions about what we should see in terms of African ancestry in non-African populations. We can put this on a continuum. So down here we have multi-regional evolution, which would say that some of the important mutations involved in becoming humans occurred in Africa, but others occurred elsewhere. And anyway, regardless of where they occurred, they would be spread around by gene flow so quickly that we wouldn't expect to see any signal of where they originally occurred. And then at the one extreme, we would have African origin with complete replacement, which would say that we're basically all 100% Africans in our ancestry. Then in between would be the assimilation hypotheses, which would say that most of our ancestry is African, but some small, maybe 1, 2, 5%, but some small but detectable amount of our ancestry should be have been contributed by these some non-African archaic hominin group. So this says if we want to distinguish among these hypotheses, we should look at the genes. And the first gene to be looked at any detail, sufficient detail to try to address these different hypotheses was this one. It's mitochondrial DNA. It's found outside the nucleus of the cells in the mitochondria. It has a number of unusual and useful properties, one of which is that it's transmitted from mothers to sons and daughters. Males have mitochondrial DNA, but they don't transmit it to their offspring. So by studying variation in mitochondrial DNA, we're learning about the maternal history of human populations. The first in-depth study of human mitochondrial DNA variation was carried out by Rebecca Kahn and myself when we were graduate students in Alan Wilson's lab. And we produced this sort of now iconic phylogenetic tree of mitochondrial DNA lineages in different human populations, where the first split in the tree separates a branch only of Africans from everyone else in the world, including some African individuals. And the simplest explanation for this pattern would be that the ancestor was African, left some descendants in Africa, and other descendants went out of Africa. There's also a scale here, the amount of sequence divergence. So this is basically an estimate of how much mutational diversity has arisen since the common mitochondrial DNA ancestor. And using a molecular clock approach, we can estimate that this amount of mutational diversity corresponds to around 140 to 280,000 years. So mitochondrial DNA indicated a recent African origin. Now, this study was met with a lot of controversy, with a lot of debate, a lot of criticism, some of it justified, some of it not. But further studies have basically upheld that same picture. So the picture we have today of mitochondrial DNA, now using complete mitochondrial DNA genome sequences rooted with the chimp, is that we get a phylogenetic tree very similar to what we saw before, and that it would take about 150,000 years to produce all of the mitochondrial DNA diversity that we see in modern humans today. So mitochondrial DNA clearly indicates a recent African origin. But mitochondrial DNA is just one gene, and we know that the single gene history can differ from that of the species because of chance events or perhaps because there's been selection. So we need to look at other genes. The next gene to be looked at in sufficient detail to address these questions was this tiny, pathetic little chromosome here. This is the human Y chromosome compared to an X chromosome. It's what makes us males male. And it's the sort of paternal analog to mitochondrial DNA in that it's transmitted from fathers to sons. So it provides insights into the paternal history of populations. And studies of Y chromosome variation in modern humans produced trees that look very much like the mitochondrial DNA tree, with primary division separating Africans from other groups, and that it was taken around 200 to 250,000 years to produce all of the Y chromosome variation that we see in modern humans today. So the Y chromosome also has a strong signal of a recent African origin. And of course, nowadays, we're well beyond these single gene sort of studies. We can look at genome-wide variation. We can look at variation across all of the chromosomes that we have. And so when we do that, we invariably get trees that look much like the mitochondrial DNA and Y chromosome trees, where the first splits involve African populations. Moreover, with genome-wide data, we can do a lot more than we can do with just mitochondrial DNA or Y chromosome data. So one thing we can do, for example, is we can use computers to carry out simulations of different population histories, different demographies, and ask what simulated population history gives the closest fit to the real genome-wide data that we observe today. And those simulated histories always, without exception, involve a recent African origin of modern humans. So it's not just mitochondrial DNA, it's not just the Y chromosome, it is genome-wide that there is a strong signal of a recent African origin. And so studies of genetic variation in modern human populations, I think, and I think most people would agree, pretty convincingly rule out multi-regional evolution. But what about out of Africa with complete replacement versus out of Africa with assimilation? And here, the studies based just on modern human populations were really not able to distinguish between these two hypotheses. You would occasionally get a study which said, oh, here's a very strange pattern of variation. This looks like this must have been something that came from Neanderthals. And then that would be followed up by a few months later by another study saying, well, actually, you can get the same pattern with out of Africa and complete replacement. So the argument went back and forth and back and forth. But we do now have an answer as to which of these is the best fit. best model for human origins. And that comes from additional data, not from modern humans, but genome sequences directly from archaic humans, so Neanderthal genomics. So the first Neanderthal genome sequence was produced in 2010. It was a very messy sequence. It was very low coverage, lots of errors, but it was sufficient to address this question of whether or not there's any Neanderthal ancestry in modern human populations. So it was a composite of DNA sequences from 3 different bones, all coming from this cave here, Vindija Cave in Croatia, and they're all dated to in excess of 38,000 years ago. Now, there were 3 different analyses done to address this question of whether or not there's Neanderthal ancestry in modern humans. All 3 gave the same result, so I'm just going to walk you through one of them, and I'll skip the gory mathematical details, but I want to give you some idea of the logic that's involved here. So one prediction we can make is that if there was admixture, then the Neanderthal sequence should match alleles in some humans that descended from the admixture event significantly more often than in others who are not descended from the admixture event. So the test is very simple. We compare 2 human sequences with different alleles, one of which is the ancestral state, one of the other which is the derived state. And we can tell which is ancestral and which is derived by looking at sequences in non-human primates. And that tells us which is the ancestral allele when we have a polymorphism, when we have a difference in the sequence, for which the Neanderthal has the derived allele. Okay. Now, if we take 3 different sequences and compare a Neanderthal to 2 humans, just 2 humans at random, say Donald and George, most of the time what we will find is that we will see that the Neanderthal has the allele and both humans have the derived allele, and that would reflect a mutation that occurred after the separation of the Neanderthal lineage and before the divergence of the human lineages. Or we might see that the Neanderthal has the derived allele and the 2 humans have the ancestral allele, and that would reflect the mutation that occurred specifically on the Neanderthal lineage. And when we take a Neanderthal sequence and 2 human sequences, and we just look for positions that are different, vast majority of the time, this is what we see. But occasionally we see discrepancies between the gene tree and the species tree. So these are cases where the Neanderthal has a derived allele and one human has a derived allele, but the other human has the ancestral allele. There's 2 possible explanations for this mismatch. One is ancestral polymorphism, that the mutation from the ancestral to the derived allele occurred in the ancestral population of Neanderthals and modern humans. And then by chance, the Neanderthal gets a derived allele, one human gets a derived allele, the other human gets the ancestral allele. The other potential explanation is admixture with Neanderthals. And so this would be a mutation from the ancestral to the derived allele that occurred on the Neanderthal branch. And then the Neanderthals contributed this derived allele to some humans, but not to others by admixture. How can we tell the difference between these 2 potential explanations? It's pretty straightforward. In the case of ancestral polymorphism, we expect the Neanderthal to match the 2 humans with equal frequency. That is, we expect Donald and the Neanderthal to have the derived allele just as often as the Neanderthal and George have the derived allele. It's basically like a coin flip, which one gets the derived allele, which one gets the ancestral allele. In this case, we would expect Donald to match more significantly often to the Neanderthal than George would. So that's the basic of the test. We compare diverse human genome sequences from around the world and ask if one shares more alleles with the Neanderthal. If we can take 2 Africans, we see no excess matching with the Neanderthal. If we take 2 non-Africans, no matter where from around the world, we also see no excess matching with the Neanderthal. But where things get interesting is when you compare an African to a non-African, then you always see, without exception, an excess of matching of the non-African to the Neanderthal. And what's interesting is it doesn't matter where the non-African is from. It could be a French individual as shown here, which would make sense because Neanderthals were in Europe, along with the ancestors of French people. But you could take a Han Chinese, you could take a Papua New Guinean, you could take someone from the New World. They all show an excess of matching compared to an African, to the Neanderthal. And so that's sort of summarized here, that we see this excess matching. Now, it turns out there's 2 possible explanations, though, for this excess matching. One would be that when the ancestors of modern humans left Africa, they fairly quickly encountered Neanderthals, interbred with them, picked up Neanderthal ancestry, and then started moving out elsewhere into the old world and diverging from one another. And so there would be this shared signal of Neanderthal ancestry in all non-African populations. But the other possible explanation is that there was ancient structure within Africa. Now, this is a little hard to explain, but the basic idea is that Neanderthals also came out of Africa several 100,000 years before modern human ancestors did. So if the African population that gave rise to the ancestors of Neanderthals also later gave rise to the ancestors of modern humans, then we would expect modern humans outside of Africa and Neanderthals to also share some excess alleles, not because of interbreeding, but just because of the shared ancestry, common ancestry. The test I just told you about doesn't distinguish between these two, doesn't tell the difference, but there are other tests one can do that do. And when applying those other tests, this explanation is pretty convincingly ruled out. And so we're left with the explanation that there was indeed interbreeding between Neanderthals and the ancestors of all non-Africans. So with the aid of Neanderthal genomics, we can rule out of Africa with complete replacement, and we're left with out of Africa with assimilation, where there's around 1 to 2% Neanderthal ancestry in non-Africans. Now, in the course of doing this work, Svante Pääbo's group, who was leading the project, the effort, were trying to get their hands on any fossil that might potentially be a Neanderthal to see if it would have enough DNA to produce a genome sequence. And one of the fossils they screened was this tiny little fingertip bone that came from this cave. This is Denisova Cave, located here in the Altai region of southern Russia, so you can see how small it is. And when screening the mitochondrial DNA, Neanderthals have different mitochondrial DNA than modern humans, and so it's a good screen to tell whether you're dealing with a human or a Neanderthal fossil. They instead got an unexpected result because it was not a modern human sequence, but it was also not a Neanderthal sequence. It was something that had never been seen before. So here's a tree of modern human mitochondrial DNA sequences. Here in blue are some Neanderthal mitochondrial DNA sequences, again, falling well outside the range of modern human diversity, diverging maybe 500,000 years ago from the modern human mitochondrial DNA sequence. Here's the Denisovan, it's twice as divergent, diverged about 1 million years ago. But remember what I said about the history of a single gene not necessarily reflecting the history of a species, and this is an example, because when you do the genome sequence from that Denisovan fingertip bone, you find out that Denisovans are actually a sister group to Neanderthals. And the divergence between the Denisovans and the Neanderthals is slightly bigger than the biggest difference that we see in genome sequences from modern humans today. So Denisovans are then this sort of novel hominin group that we knew nothing about before we had this genome sequence. It's a sister group to Neanderthals. Now we can ask, did they interbreed with modern humans? And so we do this matching statistic I told you about. Compared to Eurasians, no excess matching, a Eurasian to an African. There is excess matching of the Eurasian to the African, but it's about what you would expect given that the Denisovans are a sister group to Neanderthals. So nothing peculiar there. Where things get interesting, though, is when you compare a Melanesian, someone from New Guinea or Australia, to a Eurasian, whereas with the Neanderthal, you don't see excess matching. With the Denisovan, you see excess matching of the Melanesian, and that also holds in Africa as well. And so what it looks like there's been, in terms of a very simple model, is that there's been 2 episodes of interbreeding between archaic and modern humans. One from Neanderthals into the ancestor of all non-Africans, the other from Denisovans specifically into the ancestor of Melanesians. But if there's one thing we've learned about human evolution is that simple models are not likely to be correct. And what we know now from a decade of further research is that there have been multiple episodes of interbreeding between modern humans with Neanderthals. So here in this tree, you see the Neanderthal gene flow into the ancestor of all non-Africans. And you also see the Denisovan gene flow into the ancestor of Melanesians. But it turns out that Europeans and East Asians have a little bit more Neanderthal ancestry than Melanesians do, suggesting additional gene flow there. And the human populations that have the highest amount of Neanderthal ancestry are not Europeans, as you might expect, but rather East Asians. And so it looks like they may have also experienced a little bit of additional Neanderthal gene flow. We're also finding multiple episodes of interbreeding with Denisovans. East Asians originally thought not to have Denisovan ancestry. Now we know they have a very tiny amount of Denisovan ancestry, around 0.2%. And moreover, that seems to reflect at least 2 different pulses of Denisovan gene flow in East Asians. It's really remarkable. We also see this in Papuans now, at least 2 pulses of Denisovan gene flow in Papuans. And the most recent of these is dated to about 30,000 years ago. We're in New Guinea. And so it suggests, it's quite controversial, but it suggests Denisovans may have been in New Guinea, been able to make the water crossing to get into Australia and New Guinea. And we also see Denisovan ancestry in the Philippines. And some of these Philippine groups have the highest amount of Denisovan ancestry recorded. And it's clearly different ancestry than what we see in the Philippines. So many different pulses of Denisovan ancestry in modern humans. We're also finding other Neanderthal ancestry. So this is a fossil called Awasi 1. It's dated to about 40,000 years old, comes from this cave in Romania. It's been described as being a modern human, potentially with some archaic features. Turns out to have a whopping 6 to 9% Neanderthal ancestry compared to 1 to 2% that you would find in you or me today. And so this means that the Neanderthal interbreeding in the ancestry of this individual occurred about 4 to 6 generations prior to this individual being born. So very close in time to the interbreeding event. And even more interesting, if you look at that Neanderthal ancestry, it's not the same as the Neanderthal ancestry that we see in modern humans today. So it's an example of an interbreeding event between modern humans and Neanderthals that didn't go anywhere, that went extinct. Probably there was a lot of that went on. We're also starting to detect modern human ancestry in Neanderthals. The first high coverage, high quality Neanderthal genome sequence came from the Altai region, actually came from Denisova Cave, where there were both Neanderthals and Denisovans. This Altai genome sequence has about 1% modern human ancestry. Now, nobody thought much about that because you always have a small amount of modern human contamination in these Neanderthal fossils from the people who handle the fossils or maybe from the laboratory manipulation. But then Martin Colville, a postdoc in Svante's group, got the bright idea to, well, let's take a look at that modern human ancestry and just see what does it look like. Lo and behold, it turns out it was not related to modern humans, as you might think. Instead, it branches off prior to all of the divergence of modern human genome sequences. So it reflects a modern human population that existed prior to the out of Africa migration from which all modern populations are derived out of Africa today. So it suggests that there was an earlier migration of modern humans from Africa that contributed ancestry to Neanderthals, but did not contribute any ancestry to modern populations. And we're even detecting admixture between Denisovans and Neanderthals. Now, whenever paleontologists excavate a cave, they will end up with hundreds and thousands of very tiny little bone fragments that are not diagnostic. You have no idea what sort of creature they might have come from. And usually they're just put in a bag and put aside. Now, in principle, one could take those fragments and do ancient DNA analysis, but given the cost and the amount of time it takes to do this, it would be prohibitive to try to screen hundreds to thousands of samples. You just can't do it. But a few years ago, the method was developed to look at not at DNA, but at ancient proteins. And with ancient proteins, you can tell what family you're dealing with of mammals. And so in one of these studies, 2,400 bone fragments from Denisova Cave were screened by this method, this mass spectrometry method called ZOOMS. One of those was identified as being a hominid. So now you do the ancient DNA on that one fragment, and here it is in all its glory. And remarkably, the genome sequence indicates that this individual had a Neanderthal mother and a Denisovan father. And there's also some indication that the Denisovan father had some Neanderthal ancestry as well. So I think if you want to sum up what we've learned about human evolution from archaic genomes, it's this. Everyone was having sex with everyone else. And that's what characterizes human evolution. Now, this archaic ancestry also is a new source of information, not only on the mating habits of our ancestors, but on selection and adaptation that occurred during human evolution. So here is a cartoon of the 22 human chromosomes with the red and blue bars indicating Neanderthal ancestry found in Asians and Europeans. And what you can see is that Neanderthal ancestry is not distributed at random across our chromosomes. Instead, it's clumped. And there are some regions that have significantly less Neanderthal ancestry than expected. These are called deserts. And so presumably, these reflect regions of the genome where Neanderthal variants don't work well with modern humans, that there's something significantly different between modern human and Neanderthal genome sequences in these regions, something of functional interest. And so if you start to look at the genes that occur in these deserts, one of them is a gene called RUNX2, encodes a transcription factor that is involved in turning on expression of many other genes. And mutations that eliminate the function of RUNX2 result in a disease syndrome in modern humans called cleidocranial dysplasia, or CCD. And some of the features of this syndrome are you get delayed closure of the cranial sutures of the bones of the skull. You get frontal bossing, this protuberance around the eyebrows. You get abnormal shoulder bones and collar bones. You get a bell-shaped rib cage, and you get dental abnormalities. And what's interesting about these features is they recapitulate some of the skeletal differences between modern humans and Neanderthals. So Neanderthals, for example, have this protuberance over the eyebrow ridge called frontal bossing. Modern humans don't have it. Individuals with CCD have this. Modern humans, we have a barrel-shaped rib cage. So our rib cage is vertical, going from top to bottom. Neanderthals have a bell-shaped rib cage. It flares out going from top to bottom. Individuals with CCD have a bell-shaped rib cage. They also have abnormal collarbones and abnormalities in the shoulder bones, and these are exactly the sorts of things that you see in Neanderthals. So we've learned something then about at least several of the genes that are involved in the differences between Neanderthals and modern humans by looking in these deserts. But now, in addition to desert, there are also islands of Neanderthal ancestry. These are regions of the genome with significantly more Neanderthal ancestry than expected. And so if we look at what sorts of genes occur in these islands, we find that they occur in a couple of different functional categories. So they include genes that are involved in the immune response and how we respond to parasites and to disease-causing organisms, bacteria and viruses. They are involved in skin morphology, skin pigmentation, and they are involved in a diet, how we respond to different dietary sources. And if you think about it, I think these all sort of make sense, right? These are ways in which we have to interact with our environment. In Neanderthals, they came out of Africa several 100,000 years ago. Now they're in this new environment, new climate, new disease organisms, new sources of nutrition. They have to adapt to all these novelties, and they do so because they're successful for hundreds of thousands of years. And then our ancestors come out of Africa, and they have to do the same thing all over again. So maybe by this adaptive introgression, we picked up selective variants that were already proven to be good in this novel environment. So who knows, maybe without this adaptive introgression, maybe our ancestors would not have been able to survive outside of Africa. It's interesting to think about. So these all sort of make sense, that these would help modern humans adapt to the new conditions outside Africa, that these would be selected for. But there's some other signals that are a little harder to explain. So one of these is in a gene called EPAS1. It turns out this is a gene that's been well known to be involved in high altitude adaptation in Tibetans who live in altitudes in excess of 4,000 meters. So these colors show you the different haplotypes, the different variants found at this gene in different human populations. The TIB population is Tibetans, and you can see they have a very high frequency of this variant, which is the selected variant that works well at high altitude. You ask, where else do you find this? Hardly anywhere else in modern human populations. But it turns out that this matches the Denisovans. And in fact, it looks like Tibetans got this adaptive variant from Denisovans. Now, that seemed really hard to believe that Denisovans could be living at such high altitude. But further support for this came a few years ago from a study. It's actually not a new fossil find. It's an old fossil find that's been newly analyzed. It's a jawbone, as you can see, that came from the Spashia Karst cave, been dated to about 160,000 years old. Tried to get ancient DNA from it, but this is too old. There's no surviving authentic DNA there. But look at the proteins. The protein suggests it is indeed Denisovan. And in a follow-up study that looked at DNA from the cave sediments, there's Denisovan mitochondrial DNA in the cave sediments. So yes, it looks like Denisovans were indeed capable of living at such high altitudes, which then, makes you sort of wonder, so what were these Denisovans anyway? Could they have been the source of the myths about the abominable snowmen? So I'd like to conclude now with take-home messages. So in addition to telling us about the sexual habits of our ancestors, the archaic genomes are a source of great new insights into the dispersals of modern humans. They tell us about the genetic differences that make us human, and they tell us about alleles of functional importance that we received from archaic humans. So I'll end with the acknowledgements. The work that I talked about on Neanderthal and Denisovan genomes was the work of a great many people from a great many institutes. These are the main players at the Max Planck Institute for Evolutionary Anthropology in Leipzig, and the fearless leader, Svante Pääbo. As you probably know, Svante was awarded the Nobel Prize last year for his discoveries concerning the genomes of extinct hominins and human evolution. And I think it's just a great, great acknowledgement of the importance of ancient DNA and human evolutionary studies for not just medicine and physiology, but for evolutionary genomics and for humans in general. And of course, we also have to acknowledge the Max Planck Society for the funding that makes all this work possible. And finally, if you'd like to know more about molecular anthropology and what we've learned using molecular approaches to address anthropological questions, there is a book I can recommend. And with that, I'll end, and thank you.